Hello everyone and welcome back to Clinical Cousins YouTube channel. Today we're going to go over the process of photoreception and transduction, or simply how we see. So the first thing that we need to know is that light particles, little tiny massless particles called photons, enter our eyes and pass through our pupils where they land on the retina. As you can see right here, we have a little photon passing through the pupil. He lands on the retina. What I've done right here is I've drawn all the layers of the retina so we can break this down. So the retina is an umbrella term for the 8 or more different layers of nerve cells and their connections. So what happens is that the light strikes the photopigment layer, which activates the big photoreceptor layer, and this photoreceptor has its cell body in what is called the outer nuclear layer. Now this outer nuclear layer connects to the outer plexiform layer, which connects to cells here who have their bodies in the inner nuclear layer, which connect to the ganglion cells in the inner plexiform layer, which then connect to the ganglion layer, which then each ganglion cell has an axon that bundles itself together, where we eventually have the optic nerve layer, which is our actual optic nerve. So I know that this is a lot to grasp, but we're going to break it down today so that you understand this whole process of photoreception and transduction. So the way that I remember the layers is POIGO, or P-P-O-O-I-I, go. So first we have the pigment layer, we have the photoreceptor layer, we have the outer nuclear layer, we have the outer plexiform layer, we have the inner nuclear layer, the inner plexiform layer, the ganglion cell layer, and then finally the optic nerve layer. So a general overview is that the photopigment sits within a photoreceptor cell. So this whole thing, I've drawn a photoreceptor cell. This is not actually what it looks like. This is not actually to scale, but I've drawn this diagram as sort of a physiologic diagram so that we can understand it. So this photoreceptor cell actually has its cell body, or all of its machine parts that make it work, in the outer nuclear layer. So the outer nuclear layer. Then it sends its axon to meet with other cells in the outer plexiform layer. Remember that a plexus is really just a network of connections. So these other cells that we will talk about a little bit later, they have their actual cell bodies in the inner nuclear layer, and they themselves make connections in the inner plexiform layer to our ganglion cells, the green G ganglion cells. So these ganglion cells, what's really interesting is that they have axons, which is expected, we know this, but the interesting thing is all of these axons bundle together to make our optic nerve. So let's break this down. First, we need to know that there's a substance called rhodopsin. This is a sort of pigment that is sensitive to light. This means that when photons of light, as shown here, strike rhodopsin, rhodopsin changes its shape. Basically, light is able to bend rhodopsin into a new shape. So imagine rod, like rhodopsin. Imagine a rod that gets bent when light hits it. So first we need to know that there are two shapes of photoreceptors. Both of these photoreceptors contain rhodopsin, and these are called rods and cones. So what makes them different is that rods, based on the way that they're shaped, they're called rods. They have a ton of rhodopsin, and they're basically just bursting at the seams with this stuff. And this means that our rods are very, very sensitive to light. And this is what allows rods to be able to pick up light even in dark conditions. So this is mainly associated with our black and white vision. So next we have cones. So these shapes contain a little bit less rhodopsin and are therefore less sensitive to light overall, but they aren't useless. They are involved in our color vision. So now that we have settled that and we know the two shapes of photoreceptors, I've drawn sort of a rod shape here. We need to look at how light starts a chain reaction to eventually lead to an image going to our brain. So let's remember that the first part of our retina is called the photopigment layer. So when it is dark and there are no photons around, this layer is full of a form of rhodopsin called 11-cis retinol. So 11-cis retinol is basically just a photopigment that is in its resting position. So that's how we can think about it. Now, remember that cis, C-I-S, means same. So it's the same throughout. Now, an interesting thing happens when we enter light into the picture. So when light photons strike this 11-cis retinol, the 11-cis retinol gets bent and transforms into all trans retinol. So this is very easy to remember. It transforms into all trans retinol. So this is the activated form of our photopigment. So all of a sudden, an increase in this all trans retinol leads to the production of a very important product called Metabotropic 2. So this is a very important substance because Metabotropic 2 goes and binds itself or glues itself to a protein called transducin. So hopefully you get it. You're making associations. Transducin. Why? Because we are transducing. We are taking photons of light and we are transducing or transforming it into an electrochemical signal. So what has happened? Well, we know that the photon has struck 11-cis retinol, which has turned it into all trans retinol in the photopigment layer. Next, we know that this all trans retinol has created Metabotropic 2, which has glued itself. Metabotropic 2 has glued itself to transducin. So now something very interesting happens. So usually at rest, or we can say in the dark, transducin is just sitting there and minding its own business, while nearby a substance called cyclic GMP is bound to a sodium channel. And because cyclic GMP is bound to the sodium channel, the sodium is coming into our photoreceptor layer and our outer nuclear layer. So we remember that sodium is positive. This means it's going to be depolarizing the cell. So this sodium channel is sometimes called a dark sodium channel because it is activated in the dark. So cyclic GMP is bound to the channel and sodium comes in and it depolarizes the cell when it's dark. That is what's happened. Now, what happens in our story when we have light? Well, we know that eventually we are going to have a huge buildup of sticky Metabotropic 2 that glues itself to transducin. So when this happens, this transducin becomes activated and it starts ripping the cyclic GMP away from the dark sodium channel, and this transducin will actually turn cyclic GMP into an inactive substance called 5' GMP. So this means that our dark sodium channel is no longer activated. That means that sodium cannot enter the cell. So the photoreceptor cell body will become hyperpolarized. Well, why is this so? Because we don't have the positive sodium coming in, we never reach threshold potential, so our photoreceptor is now hyperpolarized. And in general, what happens when a cell becomes hyperpolarized? Well, we know when a cell becomes hyperpolarized, remember it gets more negative, it stops firing. Or you could also say it becomes lazy, goes to sleep, whatever you want to say, it's inactive. Well, in our case, we see that the photoreceptor cell is depolarized or activated when it's dark, when there's cyclic GMP connected to the dark sodium channel. This means that when it is dark, the photoreceptor is active and it is releasing glutamate. Now, in our story, we have been activated by light, so we know that light eventually causes hyperpolarization. This means that the photoreceptor cell stops releasing glutamate. So what does this look like actually? Well, the photoreceptor cells that we've been talking about usually release glutamate when it is dark. This is a very important concept. When it is dark, our photoreceptors release glutamate because they're depolarized. We know that glutamate is an excitatory neurotransmitter. Now, this excitatory neurotransmitter is released onto other cells in what's called the outer plexiform layer. We can see here that the photoreceptor itself has its body in the outer nuclear layer. Get it, nuclear for nucleus, or the cell body of the cell. And we can see that its axon, located right here, connects, remember, forms a plexus, a network connection to the other cell bodies in the outer plexiform layer. Now remember that a plexus is simply just a network of connections. Well, what do our photoreceptors connect to? Well, great question. They synapse or connect with cells called bipolar cells and horizontal cells, which have their actual cell bodies in the inner nuclear layer. So we have the outer nuclear layer, we have the outer plexiform, and the inner nuclear layer. So this is a very important and interesting concept. So there are two types of these kinds of bipolar cells. So when it is dark, and when the retina is not activated by photons, this is not our story, we have something called ionotropic receptors that are activated by the glutamate from the photoreceptor cell. Now these ionotropic receptors, they send excitatory messages to tell our brain, hey, there's nothing to see here. Now on the contrary, we have metabotropic cells which are inactivated by glutamate, which means that these cells do not release messages when it is dark. This also tells our brain that there is nothing to see here. Now I know that this can be a sort of confusing concept, but we're going to break this down right now. So let's get back to our story. We see that at this point, the photoreceptor has become hyperpolarized, right? We block the sodium, we've become hyperpolarized, and we are no longer releasing glutamate onto our bipolar cells. Now, when the ionotropic bipolar cells are no longer activated by glutamate, they become lazy. You can see that this guy's a little sad. So he's going to stop releasing excitatory messages. He's going to stop releasing glutamate. Now, conversely, or on the other hand, we have our metabotropic receptors. Now, when they are no longer inactivated by glutamate, they no longer inhibit, so they can send messages. Remember, this guy, the metabotropic, he was being blocked by glutamate. So now the glutamate's gone, he is free to send these excitatory messages. So now these bipolar cells, what do they connect to? Well, they synapse or connect to ganglion cells. Now, these ganglion cells basically collect all this information, and then they pass this information onto the brain via their long axons, which we call the optic nerve. So what in the world has just happened? So let's talk about this. At rest, or let's say in the dark, the 11-cis retinol is resting quietly within the photoreceptor rod or cone. Now this means that transducin is also at rest. Cyclic GMP is actually bound to the dark sodium channel. Sodium is flooding into the photoreceptor, which is depolarized. Our photoreceptor is releasing glutamate onto our usually excitatory ionotropic bipolar cells and our usually inhibitory metabotropic bipolar cells, which pass on information to our ganglion cells, which bundle their axons together to form the optic nerve, which tells our brain, hey, there's nothing to see. Now, enter light. Enter our little photons. So what happens when you shine a flashlight in your eye all of a sudden after you've been sleeping? Well, first of all, it's going to be very uncomfortable, but we know that a barrage of photons will suddenly hit the 11-cis retinol, which transforms into all trans retinol. This all trans retinol starts a chain reaction that makes Metarodopsin 2. This sticky Metarodopsin 2 glues itself onto transducin. Now, transducin is within the photoreceptor itself, and we know that transducin will rip this cyclic GMP, which turns it into 5' GMP. Because the dark sodium channel no longer has cyclic GMP to activate it, it closes. Now, when it closes, positive sodium does not come into the cell. The cell, the photoreceptor, becomes hyperpolarized. It becomes inactive. It becomes lazy. So the photoreceptor will no longer release glutamate onto our bipolar cells. Now, this is all happening in the outer plexiform layer now. So this makes our usually excitatory ionotropic receptors go dormant. They're sad, and they will not send any glutamate messages. Now, this lack of glutamate will actually make our metabotropic receptors happy. Now they're going to start sending glutamate messages. So both pieces of information are passed on to ganglion cells in the inner plexiform layer, and these ganglion cells will take this information and they will not alter it, but they will pass it on via their axons in a big bundle, which we call the optic nerve. So this information from the optic nerve travels through our brain to our occipital lobe in the back of our head, where an image is created from the millions of pixels, if you will, of light information that we are receiving. So wow, thank you guys so much for tuning in to this exciting and I think fascinating episode on the channel, and thank you for liking and commenting, and we hope that you subscribe for more content. Thank you so much.